here today, and thanks for the arm to invite me to present SparingVision to you. So SparingVision is a biotech company that was started in 2016 in France. And I have a technical problem, which is not a good way to start. Yeah. Here you go. Okay. And so our mission is to pioneer ocular genomic medicine. As I said, we were started in 2016 as a spin-off from the Institut de la Vision in Paris. And since then, we've done a lot. As you can see from this slide, which is a summary of what we have today, we have built a portfolio from one legacy asset to 6 products in the portfolio today, being 3 gene therapy products and 3 product based on the CRISPR technology from our partner Intelia. We've raised 135 million euro in Series A and Series B, and we have a cash runway with what we've raised until the beginning of 2026. Very importantly, within the horizon of the cash runway, we will be clinically validating our 2 most advanced assets, and I will speak to this asset in a very short while. The first one is already in phase 1/2. The second one will be in clinical development in 2024. We are speaking about money in this slide because what makes us very different from our peers, inherited retinal disease, and you will see that inherited retinal disease is the primary target of our program today, is that because of their mutation agnostic nature, the commercial potential of our gene therapy product is enormous and has no comparison with what you can see of the other gene therapy program in our space. Speaking about the lead, SPVN-06, it's a product which is now in clinical development and has a potential to preserve vision at whatever stage of the intervention with the program. It's a gene-agnostic AAV gene therapy program, which primary target is RP, which is the most prevalent form of inherited retinal disease. But as soon as the safety of that program is proven, the strategy of the company is to start exploring the asset in other disease, because it's not only mutation agnostic or gene agnostic, it's also disease independent. And when you will look at the biology, you will understand why. So as I said, we are currently conducting a phase 1/2. We recently announced that the first cohort was dosed safely. We are now in the second cohort of the dose escalation part of the phase 1/2. And you'll see the design of that trial in a short while. We expect clinical readout in terms of efficacy and activity at the end of 2025 for this program. SPVN-20, the second program, we like to call it the co-lead. It's 18 months behind SPVN-06 in terms of IND readiness, but with a totally different biology. Here we are aiming at restoring function, so we expect a much shorter time to read out clinically. It's another gene-agnostic AAV gene therapy program. It will be also developed at first in RP, but the idea as for SPVN-06 is, once the safety is proven, to explore the asset in other inherited retinal disease and possibly go into AMD as well. And then we are left with 4 less advanced program, another gene therapy program, which is named SPVN-30, which is basically the combination of the transgene from the first 2 gene therapy program into one single AAV product, and then the suite of CRISPR product that we are developing in collaboration with our friends at Intelia Therapeutics. Next slide is the team. I'm not going to spend too much time on it. It's fair to say that the team is complete. You will probably find some familiar faces or name on this slide, just to name 2 of them. Daniel Chung. Dan Chung was previously at Spark. He joined us 3 years ago, and at Spark he was instrumental in the development and launch of Luxturna, which obviously is very topical with what we are doing. And Mehdi Gasmi, who joined us 2 years ago as CSO and COO, and who previously was president and CSO at Verum Therapeutics, a company that I'm sure most of you know well. That's the portfolio in a nutshell. I'm going to skip this one in the interest of time. The most recent news, so from bottom to top, in April, we've appointed Joe Papa as chairman of the board. Joe was previously CEO and chairman of Bausch & Lomb. In April as well, we had 3 poster publication at ARVO. Then in May, a first oral presentation at ASGCT on SPVN20. In August, very importantly, we've released the first data with, not exactly data, but we publicly disclosed the fact that we have passed successfully the first cohort in the phase 1/2 study with SPVN06 called Prodigy. And more recently, we've selected the second target in the Intelia collaboration. I will speak a few words about that collaboration at the end. And we are very proud that recently as well, at the end of August, early September, we've been elected First 15 company, one out of 2 in Europe, and the only French company, which is giving us a lot of pride as well. So speaking a few minutes about SPVN06, the lead, as I said, it's a mutation-agnostic gene therapy program that has started clinical development in 2023, for which we expect to have a safety readout along the way, and then efficacy readout at the end of 2025. Why so long? It's because it's a slow-moving disease, and the mechanism of action of this program is to slow or stop the progression of the disease, again, which is a slow-moving disease. So this is in a snapshot the evolution of RP, and the way this disease evolves is that you start to lose your rods, and eventually you will lose your cones. And the whole theory of that asset, SPVN06, is that you are losing your cones, which are not affected by the genetic mutation, because as the rods are dying, the cones will lack support provided in a normal retina by the rods through the secretion of what we call a neurotrophic factor. And what we have in this gene therapy is exactly that neurotrophic factor combined with a very potent antioxidant enzyme. So the combination of the neurotrophic factor, which is favoring the uptake of glucose in cones, and the action of the very potent antioxidant has an effect to preserve the cones from dying. So this is really an action on the metabolism of the cone, to preserve them from dying. So that's very nice, very clean, very beautiful science that was developed by the brain of José Saël and his team in Paris over the last 20 years, and that has started clinical development very recently. And this is obviously a very exciting new development for what we believe is a breakthrough biology. I'm going to move to the design of the ongoing phase 1/2 trial, So we are in the step 1, which is a very classical dose escalation part. We are in the medium dose, and we expect to have completed the dose escalation part towards the end of the year, early next year. We have 2 sites, one in Paris and one in Pittsburgh in the US. And then once we have completed that dose escalation part, we'll move into the step 2, which, as you can see from the slide, will be a control study, not powered statistically to be pivotal, but that's the way the design was discussed and agreed upon with the FDA. So you will have 2 doses selected from the part 1, and then 1 group of naive patients. And then 12 months after the last patient has been dosed, we will look at what we have, and that will be at the end of 2025. And very likely the next in-mind trial will probably mimic the design of the step 2, but of course in a statistically powered manner. The second asset, SPVN20, as I said, another gene agnostic or mutation agnostic gene therapy product. Here we are not aiming at preserving a function that is already there, but we are aiming at restoring function, and I will explain the way we intend to do it. In terms of where this product is today, we are just starting GLP tox. We are completing GMP manufacturing, and we should be IND ready sometimes in the first quarter of 2024. Again, importantly, different mechanism of action, different biology. We aim at restoring functions, so the clinical validation of that program should be much shorter. And we expect to enroll the first patient more or less at the same time as today, but next year, and we expect to have a clinical readout at the same time as for SPVN06, again, given the fact that this biology is aiming at restoring function, so we will be able to observe that restoration much more rapidly than when you try to preserve something that needs to be preserved. The biology is even more complex than the biology of 06. The transgene is called GERK. It's a G protein inward rectifying potential channel. And the idea here is to recreate in part of the cone that you can see on the bottom, which is called dormant cones, which are cones that are still viable, they still exist, but they have lost their ability to function because they have lost, as you can see from the top of the slide, which is a normal cone, you see that you have a part which is missing in a dormant cone, which is called the outer segment of the cone. And this is where, in a normal retina, in a normal cone, where the phototransduction cascade is happening. So in a dormant cones, which is a status that all cones go through at one point in the evolution of inherited retinal disease, and especially RP, the idea with GERK, that transgene, is to recreate that phototransition cascade within the inner segment of a cone. And you have all the ingredients like opsin, arrestin to do so. We are just missing the signal that we will be sending with that GERK transgene. So it's a beautiful technology, again, totally mutation agnostic, disease independent, as long as we need to reactivate function in cones. Theoretically, this kind of biology should be able to work. This is very often something which is compared to optogenetics. You may be familiar with optogenetics, except that here we have a transgene of human origin, and we are targeting the cones, unlike other optogenetic solutions which are targeting cells in the retina, which normal function is not to be useful for the vision. I'm going to skip the data just to say, and this is what we published at AGCT earlier this year, SPVN-06, the first product, is a subretinal injection. We've made the choice for the second product, given the fact that at least the first population of patients we are going to treat are much more advanced patients in the late stage of the evolution of the disease. Their retina is already very fragile, so we had to find a solution to inject the product that was less traumatic than the subretinal injection. So we went with what we call an IVT, intravitreal capsid. And at AGCT this year, we've proven, as you can see from those two images, especially the one on the right, that we had a fantastic distribution of the GERK protein within the environment of the foveal cone. And you may know or you may not know, but the end stage of the evolution of the disease, most of the cones will be around the fovea. So that IVT injection of the GERK protein was exactly what we were looking for in terms of the part of the retina we wanted to transduce. So that was a very important moment of truth for us, and this is what has led us to what has followed in terms of preclinical development of the product. And again, we are now starting GLP tox study with that program, and in parallel, we are putting the final touch at the GMP manufacturing. We should be ready for IND, as I said, in early 2024. Most likely the clinical trial will be conducted in Europe, and we'll see where do we go after that first in-man study. Again, probably the next step will be more or less at the same time as for SPVN-06. It will be a control study, probably starting in 2026. Last but not least, our collaboration with Intellia Therapeutics. So that deal was announced actually 2 years ago in this conference. We are very fortunate and very happy to in-license the CRISPR toolbox, in a way, from our friends at Intellia Therapeutics. It's not a typical small biotech, big biotech deal in the sense that we in-license the product. We didn't license anything, or we in-license the technology, not the product. And the way the contract works is that we have a list of reserved gene targets, and we, SparingVision, decide, we have a period to decide, to elect up to 3 targets for development of a CRISPR-based product. So far, we've elected 2 targets. There is one that we expect to be selecting within the next 2 years. Once the target is selected, we sit down with our friends at Intellia to design the best possible product concept based on their toolbox, and obviously their input is paramount in that process. Once that design is agreed upon, then Intellia goes to their lab, they do the work, we pay for it, and once the CRISPR reagent is validated, they send it to us, we put it into an AAV or any other vehicle for that matter, and we move the product forward. So this is a very exciting development for us, and we are more than happy to be collaborating with whom we believe are the world leader in vivo genomic medicine. So fast forward, what will be the next step for us? As I said, we are fully financed until the beginning of 2026, and I like to respond to the question as to what is next with the following slide, which is the last slide for my presentation today. Well, by then, hopefully, obviously, importantly, we will have one, possibly two clinically validated product, a gene therapy product, first in class, with a very clear path to BLA submission. We will have, hopefully, still very active and very live at that time, an exclusive alliance with, again, the most advanced genome editing platform in the world, being our friends at Intelia. We will have a development engine capable, and this is our goal, of bringing one new product, whether it's a gene therapy or a CRISPR-based product, to IND readiness every other year. We will have an established platform of enabling technology. I didn't have time to speak to it today, but in addition to developing our program, we are developing our in-licensing technologies that will support the portfolio, support the lifecycle management of our program. And last but not least, you've seen the team. You have not seen our KOL, but we have a fantastic network of KOL. We are building the IP platform, and so we will have all the ingredients by then to succeed and become a successful, very vertically, integrated leader in the world of ocular genomic medicine. And with that, I'm going to conclude and close my presentation. Thank you very much for your attention.